All right, so today's plan is to finish off chapter 13, which is glycolysis, and possibly start chapter 14, which is the citric acid cycle, and see how one leads into the other. So let's see here. We left off using phosphoglycerate mutase. Let's see where I can find that slide. There we go. So we left off there on our reaction, where we converted our 3 PG into 2 PG, and we had a net G of 0 because it's all in equilibrium. So now we're going to take that 2 PG and dehydrate it and turn it into phosphoenol pyruvate. So 2 PG is dehydrated. In order to do this dehydration, we're going to need magnesium 2+. Actually, let me come back to this sentence. Sorry. I'll come back to that. I want to do something else first. Otherwise, it'll seem confusing. Not that it's not already confusing. I've already stopped this like 2 seconds in. All right, so here we had 2 PG, and we're going to dehydrate. So we're going to remove a water and make ourselves an enol, which was, right, it's this it's carboxylic acid group followed by a double bond. Enol, and it looks similar to pyruvate, but it's phosphorylated, so it's phosphoenol pyruvate. This reaction again has a delta G of approximately 0, which what does that tell us about the directionality? Right, it's equilibrium. So in this case, there's no preferred final product. So we're going to have to wait for the next step to force the reaction to continue. So to take that a step further, let's say we have 2 PG building up in the system, like we're not needing any PEP yet, so we have this building up. Our previous step was also an equilibrium step. Right, that was also equilibrium, which means that our original 3 PG would also build up. So all of these steps are in equilibrium, right? Making a PEP is in equilibrium with 2 PG, which you could even say is in equilibrium with our 3 PG. So all those steps are in equilibrium with each other. Okay. So this fluoride ion is an inhibitor of this reaction. And it's because enolase needs magnesium +2. So what have we used magnesium +2 the other 3 times we've used it? It stabilizes which group? Which functional group on this? The phosphate. Yep. So same thing, enolase needs a magnesium +2 to keep the phosphate from leaving, because it's a much better leaving group, right, than water. That phosphate group would be a high energy cleave, and it'd be preferred. And so it needs magnesium to keep that phosphate group from leaving. So, let me change that. So if we have a fluoride ion, so in the presence of a fluoride ion, so if you have too much fluoride in your system, the fluoride would bind preferentially to that magnesium 2+ and keep the 2Pg from entering the active site. So that's sort of inhibition that most likely be called competitive. This would be competitive inhibition. It's not binding irreversibly. It's just preventing, depending on concentration, which one is going to be in the active site. All right, any questions on making PEP? No? Okay. Let's take PEP and make it into pyruvate. All right, so our last enzyme of glycolysis is pyruvate kinase. What do kinase enzymes do? They either take off a phosphate. So what would kinase be doing in this case? This is our final product. It's removing a phosphate group. Good. So again, kinases add, remove phosphate groups. In this case, it's removing. So PEP is going to be cleaved using pyruvate kinase to form pyruvate. If we are going to cleave off that phosphate group, what are we going to add it to? ADP. So you know in this reaction, if there's a kinase, and it's at the end of the reaction, you're going to remove a phosphate group, which means there better be some ADP, which there is, around so we can make ATP. This is a two-step enzymatic reaction. So in the first step, we're going to have ADP, which is shown here, with my magnesiums, holding onto that charge. So I got my Mg2+, which I just made totally illegible. Here we go. You can see it from before, and I wrote on top of it. All right, so we got Mg2+, which is holding onto those ADPs to hold those charges to keep the phosphate groups from cleaving anymore. This ADP is going to nucleophilically attack the phosphate group on the PEP, which is going to cleave it off, and we'll get our ATP. What bond is this? Has one name, starts with a P. Phosphoanhydride. Yep, phosphoanhydride bond. So it's going to cleave that phosphoanhydride bond. So now we have, what we had was phosphoenol pyruvate, so now we've gotten rid of the phospho, and so now we just have an enol pyruvate. So in step 2, we are going to tautomerize to pyruvate, which means we just rearrange the positions of those double bonds. Right, so in this case, I have a double bond to an oxygen and a double bond to a carbon. I'm going to rearrange where those double bonds are and move the double bond to this oxygen. So that one's going to stay, and I have a double bond to an oxygen. So, looking at this mechanism, do you expect this delta G to be positive, negative, or 0? We have cleaved a high energy bond, and we've made ATP. So is that likely to go this way? Is it likely to go, so if I say it's a negative delta G, it makes products. It's positive delta G, it go back towards reactants. If it's 0, it means it can equilibrium back and forth easily. This should be a negative delta G. So if you go back and you look at all your kinase reactions, those should all be your high energy reactions. Either because they're adding a phosphate group, so it's going to be positive delta G, or removing a phosphate group to make ATP, which would then be negative. So if you look back, this is actually super negative compared to a lot of the other delta Gs we've looked at. What else about this reaction? ATP, PEP. This reaction is the end of the second half of glycolysis. At this point, we've made 2 pyruvates. And 4 ATP. How did we make 2 pyruvates? Because I've only shown 1. Yeah, it happens twice, because at step 5, right, we broke our 6 carbon into 2 3 carbons. So each of those 3 carbons can now come through the system. So for every 1 glucose, I make 2 pyruvates and 4 ATPs. How many ATPs do I need in the first half? 2. So net overall, I only gained 2 ATPs because I needed 2 in the first half. So I get 4 in the second half, but overall, 2. Good. So let's look at the overall glycolysis process. So we make 3 products in our glycolysis system. This is looking at the second half specifically, starting with GAP. So in the end, we make 3 overall products. We make ATP, so we invested 2 ATPs in our first half. We generate 4 ATPs in the second half. Which makes our net yield overall 2 ATPs. We also produced NADH. So anytime we do a glucose oxidation, we do 2 NAD pluses reduced to, that should be 2 NADHs. And so it's an oxidation reaction, so we see that there's going to be some electron transport going on, which we'll need when we get to citric acid cycle. And finally, we make 2 molecules of pyruvate, which will be used in the citric acid cycle to complete the oxidation all the way down to carbon dioxide. So we'll need those pyruvates immediately. So the net reaction So in the first half, we put in 1 glucose, we put in 2 NAD pluses, and we put in, 2 ATP, 2 NAD pluses, 1 glucose. We get out 2 pyruvates, we get 2 NADHs, which also come with 2 protons. And finally 2 ATP. Questions on our net reaction? Okay. Then let's take a look at what are some possible next steps. So either our pyruvate can head down to the citric acid cycle, which is next chapter, which is the majority of the pyruvates can head down to the citric acid cycle. Or we can do homolactic fermentation, or we can do alcoholic fermentation. So pyruvate is a common intermediate that can go down 3 different pathways. So it's a common intermediate of 3 different pathways, and depending on the needs of the body and the ability or the concentration of oxygen at the time, one of these 3 pathways will be chosen. So under aerobic conditions, which one's aerobic? With oxygen, without oxygen? This is with oxygen. If there's oxygen present, pyruvate will head down the citric acid cycle. Under anaerobic conditions, pyruvate is converted to produce more NAD+, which can make a few different byproducts. So if there's no oxygen, our pyruvate will be converted to, in your body, lactate, in order to reoxidize the NADH, which was produced in the GADPH reaction. So it's going to feed back. It reduces the NAD back to NAD+, so it can go back up into the glycolysis cycle. So, So in human muscle tissue, pyruvate under anaerobic conditions makes your NAD+ and lactate, which we'll talk about here in a minute. In plants, Pyruvate can be decarboxylated to make carbon dioxide and ethanol. And NAD+. So let's take a look at homolactic fermentation first. We'll look at the other one. Everybody have it? I'll wait. Okay. So let's take a look at your body when the demand for ATP is high, but the supply of oxygen is low. So this is most often when you are climbing the stairs, spiral stairs to get to the third floor, right? Take the elevator. Yeah, those stairs are killer. I don't know how they designed them to be exhausting, but they are. So your need for energy is high, but you're not getting enough oxygen into your system. We can generate ATP by what's called anaerobic glycolysis. That's not spelled right. There we go. There's a method to rapidly generate ATP, but it's going to require a different enzyme than we use in regular glycolysis. We're going to use lactate dehydrogenase. To catalyze the oxidation of NADH by pyruvate. So as you can see over here, we have pyruvate, right, which has our carboxylic acid group, we have a ketone, and then just CH3. We're going to oxidize that ketone to an alcohol. And by doing that, that's going to turn my NADH into NAD+. Right, so just a regular oxidation reduction reaction. So I've oxidized pyruvate, and I'm going to reduce NADH+. What do these double arrows tell me about this reaction? What's my delta G most likely? It's going to be zero-ish. So this reaction is freely reversible, which means that delta G is about 0. That I can make lactate, but when I need more pyruvate, it is easy to convert back to pyruvate to send it down one of my other pathways. And you'll see sometimes it's referred to the 11th step of glycolysis because it's easy to go pyruvate down to lactate. And if that is truly the 11th step that your body actually wants to go down this pathway to make lactate, the anaerobic here's what we just had 2 slides ago. No, set the, just use those up. Oh my gosh, messing it all up. That should be 2ADP plus 2PI ATP plus so if your body goes down the anaerobic pathway, we still have the glucose plus the ADP and the 2PI. The NADH that was there before just got reduced. They cancel out on both sides. And now I get 2 lactate. I get some more ATP back. Actually, that'd be, yeah. I get some water and some protons. And that's what caused the lactic acid buildup when your muscles are burning. It's because this reaction produces free acid. That it actually is burning. It's the pH in your body's changing a little bit, just at that point. But it's freely reversible. So when your body gets enough oxygen, we head back this way, back up to pyruvate, and that pyruvate heads back down the regular citric acid cycle. You have a question first? No? No? No. Okay. In that case, let's take a look at plants, because I know everybody in here is a big fan of plants. Because you guys are all loving plant stuff, right? All right, you can go ahead and skip this. Go ahead and cross that out. I was going to talk about it, but it doesn't really add anything to the story. So, alcoholic fermentation. This is how, if you're like the 4 of you that's old enough to have the home kit, brew kit, this is how it would work. So, your glucose, especially using yeast, can go down this new pathway under anaerobic conditions and produce ethanol. So pyruvate, using pyruvate decarboxylates and makes acetaldehyde. And carbon dioxide. Which I know is very hard to buy unpasteurized apple cider anymore, but if you ever make it yourself, like just by squishing apples, you can also let it sit in your fridge, and when it starts to foam, you know you've now made hard apple cider. So the foaming is the carbon dioxide being produced. There's a little bit of ethanol in there too. Fun fact. So now we have NADH. So if I have NADH in the presence of alcohol dehydrogenase, what sort of reaction is acetaldehyde to ethanol? Oxidation reduction, yes. So step 2, if you see an NADH, means there's an oxidation reduction reaction. So I had acetaldehyde, which is an aldehyde. Aldehyde going to an alcohol. Is that an oxidation or reduction? If you don't know that, maybe you know the other one. Is NADH going to NAD plus? That's an oxidation reaction. So an aldehyde going to an alcohol should be a should be reduction. You should be able to, you should be able to pick those out by eye for sure. 'Cause just about, you definitely, if you don't know what the molecules are going to, you should be able to pick out at least NADH going to NAD plus is an oxidation. It's the other way is reduction. Hint, hint, if you don't know that, maybe star that and go back, study it. It'll be on the test.
On the previous slide,
Mm-hmm.
the molecular composition,
Mm-hmm.
where did we say the NADH to NAD plus is?
I thought,
That's
yeah,
where I don't know.
yeah, that's right.
Oh, wait, no, I have to.
Let's see here.
I have pyruvates oxidizing and NADH is producing.
Yep, you're right, sorry. I said it, but I wrote it down backwards. Sorry, out loud I said it right, and I wrote down backwards. Yeah, because pyruvate is going from a ketone to an alcohol, so that's a reduction. NADH, yeah. I must have said that out loud, right? And then I wrote down something crazy. It happens. Trying to talk and write at the same time, if you guys have never tried it. Very hard, turns out. Very hard to write and talk at the same time. All right, so in step 2, we're going to do our reduction of acetaldehyde to ethanol and our oxidation of NADH to NAD+. So then we have our NAD+, we're ready to send back up into glycolysis again. We can reuse the NAD+, the GADPH, or any other spot. That's most likely will be used. The other thing is this TPP, which we will talk about more in about 5 minutes when we start citric acid cycle. But TPP is an essential cofactor of pyruvate decarboxylase. TPP is a cofactor that's synthesized from vitamin B1. And TPP is going to be needed in this reaction I guess, yeah, proteinate. Proteinate.
Okay.
Proteinate.
Is that carbon, protonated carbon?
Mm-hmm. So it's needed to protonate the carbon after the decarboxylation. So the decarboxylation step is this first step, right? We remove this last carbon over here. And if we just remove that, we'd have a C, we'd have a carbanion, which is not favorable. So TPP comes in and provides this proton to make acetaldehyde, to make it a more favorable intermediate, which we'll spend a good bit of time talking about PPP and all of its awesomeness in a minute. All right, questions on alcoholic fermentation? Why your alcohol has ethanol and carbon dioxide in it? It's all bubbly. Okay. In that case, last thing to talk about glycolysis is how do we control glycolysis? More important, how does your body control glycolysis, not you personally? So glycolysis operates continuously in just about all of your tissues. But obviously your tissues don't need a ton of ATP all the time. So how do we control how much ATP is being made compared to how much you actually need? That control is called flux. So flux, if you can think about like a flow chart, so flux is depending on how much you put in, how much actually makes it down to the end. There's different like control valves along the middle that we can speed things up, slow it down, siphon it off to different spots. So how do we determine the flux through this cycle? We determine flux by looking at what are the rate determining steps of the pathway. And how do we know it's a rate determining step using delta G? Are they positive, negative, or zero? They'd be positive. Rate determining steps are going to be typically positive. And once you've kind of narrowed down your rate determining steps, you're going to want to see, do they have allosteric modifiers, which means do they have inhibitors or activators? Right, something that helps control the enzyme's productivity besides just concentration. So let's take a look at the enzymes of glycolysis. There are only really 3 good choices that have either large negative, which means they're irreversible, right? That's what we talked about before. If they have a large negative delta G, irreversible, or if they have a large positive delta G, they'll need to be activated in some way, right? That's how we looked at rate determining steps. So we're not going to look at delta G 0 prime in this case because that is ex vivo. We want to look at the in vivo. It's another discussion for another time. But we're going to look at this column. There are 3 reactions of glycolysis that can potentially control the whole system. All right, we got hexokinase. We have phosphofructokinase. And we have pyruvate kinase. So those are the 3 enzymes that have a large negative delta G in the actual system. So we can logically think through which one is most likely the overall rate determining step by thinking of like this is a funnel, right? So hexokinase is the very first reaction. And then we have pyruvate kinase, which is the very last reaction. If you're thinking about a funnel trying to get to the end, would you want the stopper at the top and bottom, or would you want one kind of in the middle where you can put things off to the side? The middle. You would prefer to have a stopper in the middle so that you can control how much is going in, how much is going out. Because at the top, and you turn it off, well, it's going to spill out the top. If you have it at the bottom, then you don't have good control over all the reactions in the middle. Which means that our phosphofructokinase is the overall which we could also think about. Phosphofructokinase is the one that has all the other allosteric mechanisms on it, right? It can be activated by more ADP. It can be deactivated by using phosphofructokinase 2 or ATP. So it has the most regulation as well as being in the center of the cycle. So all those are good reasons that is the overall rate determining step. How do we feel about glycolysis? Pretty excited, right? So, I'm just going to give you an introduction to the citric acid cycle. And we'll spend many, many more days talking about it. Because briefly, look how easy that is. That's what I'm saying. Pretty obvious what goes in, what comes out, what all the intermediates are, what the activators and inhibitors. Yeah. I mean, it's only 8 steps. I mean, glycolysis was 10 or 11, depending on how you're counting. Clearly just 8. I mean, altogether, just 18 easy steps. Glucose to carbon dioxide and water. Okay, plus another 4 to get us all the way through electron transport. So 22 easy steps. You should be able to know the name, be able to tell me a little bit about the structure, the enzyme, what goes in, what goes out, energy steps, overall energy.
So where's the loophole?
No, second semester they assume you already know this. They do a whole bunch of other stuff.
So it's chapter 11 and chapter 12, right?
Second semester. Chapter 11 and 12? Wait. Yeah, we're in,
No, 11 and 12.
oh, 11 and 12. Sorry.
That we just covered.
Yeah,
Well, I mean, that's planning on being on the exam.
Mhm.
That's gonna
yeah.
touch on it.
It's just going to touch on it. Like carbohydrates, it's just going to be like, what's the difference between an aldose, ketose? What's D versus L? You know, what's an alpha 1,4 versus a beta 1,4 linkage? That's about it. In chapter 12 is mostly just what we covered. Oh, the things in chapter 12 that's most important, the structure of NAD, FAD, ATP, enzyme CoA. Not draw them, be able to identify them. There's going to be a lot of other things you can draw. I just want you to be able to recognize those 4 big cofactors. NAD, FAD, CoA, ATP, ADP.
But the bulk of it, knowing it in and out, is
13, 14 is going to be the majority of it.
Our quiz this week
Quiz on Friday? Yeah.
is going to cover
I think just mostly glycolysis.
up to today. not-
Yeah, not Wednesday. I haven't decided yet. Haven't decided.
12, but it's
Where's the loophole?
NAD, FAD, CoA, ATP. There's like 3 structures in chapter 12. 4 structures. Yeah. All right, so I just want to go over this and then we'll spend Wednesday, Friday, most of next week going through this. So, as our introduction, there's 8 reactions plus 1. Because officially the citric acid cycle starts with acetyl-CoA. But we have to do the step that ends glycolysis, which is pyruvate, to get us to acetyl-CoA. So there's really 9 reactions in the citric acid cycle. 8 that are acetyl-CoA back to acetyl-CoA. There's 1 step that we've already looked at a little bit. This step right here, there's the +1, because we're going to use our enzyme that we just learned about, pyruvate dehydrogenase, to go from pyruvate to what we, like if we look back 1 slide, right, in your homolactic fermentation, wherever that went. I'll find it. It's somewhere in here. Everyone else can look back while I'm finding. Oh, here we go. Here we go. All right, pyruvate decarboxylase to acetylaldehyde. We're going pyruvate to acetyl-CoA. So we're going to use the same enzyme, because we're going to do the same decarboxylation, but we're going to add this CoA portion to it. which is a high energy bond, which we'll use later to cleave off and give the cycle a little more oomph to swing through. So we're going to send in one acetyl-CoA. We're going to get out 2 carbon dioxides, 3 NADHs, 1 FADH2, and 1 high energy compound, which is found down here. Which is GTP. Very similar to ATP, just using a different nucleotide base. So instead of nucleotide A, we're using nucleotide G. So similar in structure. All right, I'll leave everybody on that cliffhanger. Super excited. I have your homework and quizzes to pass back before everybody goes running off.